Hey guys, I hope you had a nice weekend. Today we're going to do some genetic crosses, which is Punnett squares. And I know some of you were very excited about doing Punnett squares. Most of you were probably not excited about it. I love Punnett squares and I'm kind of sad that we're not there because I had so many activities planned that we're not going to be able to do online. So this is just what a Punnett square looks like. To give you a refresher, you probably did these back in 7th grade. I don't know if you touched on them at all last year. A genotype. You've probably heard this word before. It's the alleles that an organism inherits from the parents. So there's dominant and recessive alleles. Normally dominant is going to be represented by 2 capital letters, recessive is going to be 2 lowercase letters, and you can have the heterozygous form, which is 1 of each letter. A couple prefixes that you should know. Homeo or homo, which means same, and hetero, which means different. So there's 3 different possibilities for genotypes when 2 alleles are present, and we're just going to talk about 2 alleles instead of having 3 or 4 alleles for right now. We're trying to keep things simple. So if we have 2 capital letters, we're going to call that homozygous dominant. We're saying it's homozygous because they are both the same type of letter. They're both capital, and dominant is going to be based off of that capital letter. If we have 2 lowercase letters, that's going to be homozygous recessive. And the last one is 1 of each, 1 capital, 1 lowercase, and that's heterozygous. Prefix hetero means different, so we have 2 different letters. Kind of seeing where this is going? Phenotype is going to be an organism's appearance based on the alleles that it has. So this is what you can see visibly. It would be flower color, or if you're looking at you guys, your hair color, your eye color, if you have freckles or not. A fun fact here, red hair color is actually a mutation that's based off of, I think it's the MC1R gene, and it's not actually a dominant or recessive trait. So if you have red hair, which I kind of have a hint of red in my hair, you have a 50% chance of passing that on to your child, and the brightness of that red is going to depend upon the other person's hair color. So if you have a baby with someone who has blonde hair, your baby might have a redder hair, brighter red. If you have a baby with someone who has really dark brown or black hair, then that red's going to be a little more muted, but you'll probably see if they go out in the sun, it might brighten up a lot. We got to do a little bit of math in this section, and I'm not going to have you really answer a whole lot of probability questions in the reviews, but just to refresh your memory, you should have done probability probably in 8th grade. It's the likelihood that a specific event will occur, and a lot of times you do probability with things like marbles. I remember doing those problems when I was in school. So probability is the number of times an event occurs over the total number of events. So for example, we're working with big numbers here, just bear with me, it's pretty easy. In Mendel's experiment, he counted the colors of pea pods, and he found that 6,022 pea pods were green and 2,001 were yellow. What is the probability that a dominant trait will appear in a similar cross? So he had a lot of pea plants. That's what we're getting from this whole thing. Dominant trait, that's going to be the one that shows up most often. So green showed up a lot more than yellow did, so green is going to be the dominant trait here. So we have to calculate the total number of events first. In order to do that, we're going to take our green pea pods and we're going to add it to our yellow pea pods, and that's going to tell us that there's 8,023 pea pods altogether. It's a lot of pea pods. To do probability, we're going to take our green pods divided by the total pods. So if we look at our numbers, that was 6,022 divided by 8,023, and that is 0.75. Then we want to make it a percentage, so we're going to take 0.75 times 100, that gives you 75%, or you can move the decimal place 2 points to the right, and that gives you your percentage as well. One more example. In Mendel's experiment, he counted the height of the pea plants, and he found that 787 were tall, 227 were short. What is the probability that a recessive trait will appear in a similar cross? So we got to calculate the total number of events again. We have 787 plus 277, that's going to give us 1,064 total plants. So we're looking for short plants over total plants. So we take 277 divided by 1,064, and we get 0.26. Then we're going to take that number and multiply it times 100. So 0.26 times 100 gives us 26%. Kira, this is Kira. She's one of my cats. Sweetheart, this is not the best time for pets. Sorry about that, guys. So a monohybrid cross is a cross in which only one characteristic is going to be tracked. The offspring of a monohybrid cross is just called a monohybrid. And a Punnett Square is the tool that we use to determine what the traits could potentially be. So for example, if we're looking at flower color, it's passed on through complete dominance. That means you have two completely separate traits. You can definitely tell that one is purple and one is white. So we're crossing a homozygous dominant flower with a homozygous recessive flower. In the problem, I tell you what is dominant and what is recessive. So we have our purple flower and our white flower. I know the purple flower is a cartoon, but it's the best I could do right now. So I'm going to use two capital R's because I said they were homozygous, and two lowercase r's for the white flower. And there's my square. And I'm going to put both of those letters right next to each other. So I have my purple flower up top, and my white flower is going to be down the side. And then you're going to bring those letters down and across. So you end up with two letters in each box by the end of it. So there you can see how those letters kind of combine, and that would be what your offspring would look like. We'd have one capital letter and one lowercase letter for each one. So then we move on to finding genotypic ratio and phenotypic ratios. So here is our Punnett Square. If we're looking at the genotypic ratio, we're going to start with homozygous dominant. We don't have any that have two capital R's, so that would be 0. Then you do heterozygous. We have 4 that have one capital, one lowercase, and then we have none that have two lowercase r's. For phenotypic ratios, we have two phenotypes. It could be purple or white. Because we have at least one dominant letter here, one capital letter, they're all going to be purple, no white flowers. Now, there are other forms of inheritance aside from just complete dominance. So with complete, you have two definite colors. Incomplete is where these things are going to be blending together. And that happens only in the heterozygous form. So for example, if we had two capital B's, which was blue flowers, and two lowercase b's, which would have been yellow flowers, and we mixed that together. Blue and yellow are going to make green if you remember back to art class. So let's try that then. We have red flowers are dominant to white flowers, and it's inherited by incomplete dominance this time, so we're actually going to have 3 possible phenotypes, red, white, and if you mix those two together, hopefully you know you're going to get pink. So what was the genotypic and phenotypic ratio of a flower cross for homozygous recessive and a heterozygous flower? So homozygous recessive, that's going to be 2 lowercase r's. That would be a white flower. Heterozygous, that's 1 of each. And if we mix red and white, we are going to get pink. So here's my square. You want to put both of your letters next to each other, so we'll fly those on top. Our pink flower then is going to go down the side, and it does not matter if you were to put the white flower on the side and the pink flower on top, just as long as you keep those letters together. You don't want to be splitting it and have one white flower live up here and the other one down here. That'll mess up your cross, so make sure they stay next to each other or one on top of the other. Then you're going to move your letters down and over. If we do our genotypic ratio, we're going to start with our homozygous dominant, then we'll do heterozygous, then homozygous recessive. So we don't have any that are homozygous dominant. We have 2 that are heterozygous, and we have 2 that are homozygous recessive. If we look at our phenotypic ratio, we have 3 phenotypes. So red is going to be this homozygous dominant, and we don't have any that are red. The next one is going to be the heterozygous form, which is pink. We have 2 of those, and lastly we have 2 white ones. The next type of dominance we have is going to be codominance, and this is where both alleles kind of duke it out with each other, and they're both expressed in the heterozygous form. So you don't actually have a recessive trait when it comes to codominance. If you have ever seen Monsters, Inc., and you know the character Sully, how he is blue with purple spots, he's an example of codominance because he has two traits coming out at the same time. So we're going to use two different letters here because I don't want to do a capital and a lowercase letter since nothing is recessive. Everybody's dominant here. So the red color in horses is going to be represented by two capital R's, and I say red, it's more like a brown color. White is going to be two capital W's. The heterozygous form, RW, is going to be a roan horse, which is a white horse with red spots. So there we have our roan horse. So our example here, we have horse colors inherited by codominance. When a red horse and a white horse mate, the offspring has a white base color with red spots. It's called a roan horse, with a genotypic and phenotypic ratio of offspring with a red horse and a roan horse. So our red horse is going to be two capital R's. There's our red one. And our roan horse is going to be one of each RW. So there's our spots. And we're going to keep our red horse alleles together. We're going to fly those down to the top. And our roan horse is going to stay together as well. RW goes down the side, and then you're going to just push those across and down. So you have two letters in each box. Genotypic ratio, we got to start with two R's. Normally you just go alphabetically, so R comes before W. That's why I would do the two R's first. We have two of those. Then we're looking for the heterozygous. We got two of those. And last would be that WW, the white horse. We don't have any. Phenotypic ratio should be exactly the same as your genotypic ratio. So you got two reds, two roans, no whites. Another example of codominance is blood type, and blood type is a little bit crazy because now we have three alleles instead of just two. I'm not going to have you do any of these examples, but I'll go through it just in case you're curious. So a person's blood type is going to be made up of either A, B, or O alleles. If you know what your blood type is, you can kind of figure out what your parents might have been, or if you know what your parents' blood type are, you may be able to figure out what yours is if you don't know it. The alleles A and B are equally dominant, so those two are codominant. The O allele is going to be recessive, so we have kind of a mix with the different types of dominances that we exhibit here. A person with A blood can either have two A's or they could be AO. So you still have two alleles, but it just depends on what those two alleles are. It can change. So a person with A blood can only receive blood from someone who is A or someone who is O. And you can have this AO blood type and still be considered A because the O is negative. There's really, not negative, recessive. There's no real way to determine what your two alleles are. So if you know you're A, you can't determine if you're AA or AO. I don't know of a test that will give that to you. A person with blood type B could either have two B alleles or they could have a B and an O allele. A person with AB blood only has one option. They have one allele that's A and one allele is B. And again, both of those are dominant. And a person with O blood type can only have one genotype. They are going to have two O's. So in order for you to be O blood type, both of your parents had to have one O. So here, this is just a chart showing you kind of the mix-up of our population. Roughly 40% of people are going to be A, 10% is going to be B. AB is 4% and O is 46%. It's actually pretty popular, which is kind of crazy considering that it is the recessive form. And on your blood cells, you have these little antibodies, and these antibodies are going to be attacking other cells. So that's why if you have type A blood, you can only receive from A or O. If they put B blood into you, your antibodies are actually going to attack it. So it will hurt you. You can't just get blood from anybody. I think most of the time in hospitals they try to keep O on hand just because anybody can get O blood, because if you look at it, there are no antibodies. So anybody can get it. This is a little bit easier. It shows you who the donor would be and who the recipient would be. O is the universal donor. They can give to everybody, but the downside of being O is that you can only receive from somebody who is O. A can give to A's and AB. B can give to B's and AB. And AB is called the universal recipient because they can get it from everyone, but they can only donate to themselves. They're stingy with their blood. So if we do a little example, we have a husband who has type O blood and his wife is AB. What are the genotypic and phenotypic ratios of their potential offspring? So the husband only has one option. He's OO. And the wife is going to be AB. We want to keep the husband's alleles together, so we're putting him on top. We're putting the wife down the side. Again, you could have put the wife on top if you wanted. It doesn't matter. And when we mix their blood types together here, that is what we come up with. So for our genotypic ratio, we have 2 that are AOs, 2 that are BOs. Phenotypic ratio is going to be 2 A's and 2 B's. Pretty easy. All right, guys, that's it for today. Have a great day, and I'll talk to you soon.